is Mario Salguero.
I'm Victoria Ayala.
I'm Melody Garcia.
I'm Kalina Williams.
I'm Gabby Halasi.
I'm Diana Perez.
I'm Maria Ramos.
And our project is eliminating the peanut allergy using Cas9.
So our topic is going to be eliminating peanut allergens. Why this matters is because peanut allergy is one of the most severe and persistent allergies in the world. An article by Zhuang and Perkinson in 2013 says that reactions are more particularly common and severe, and about 80% of patients stay allergic going into their adulthood. A small exposure can trigger anaphylaxis. This can be very hazardous, especially in schools, restaurants, anywhere where peanuts are like exposed to patients. So why genetic engineering is needed? Avoidance is really the only reliable option in this case, since there is no cure. So the only possible way for like a person with the peanut allergy not to get an allergic reaction is to not avoid it at all, instead of having like small doses. And then peanuts contain many proteins but not all of them cause those allergic reactions. So research shows that the RAH2 and the RAH6 genes are the most potent, and they cause the strongest IgE mediated reactions. And these two alone can change the allergenicity. They don't change the allergenicity much, but together they cause a major drop in allergenicity, and that's why these two are the most important to be removed from the allergen.
Agrobacterium-mediated genetic transformation is a genetic strategy that will help us deliver our desired peanut gene. This method will allow us to introduce our DNA, which will be our plasmid or vector, into the peanut plant, selecting and targeting our ARAH1, 2, 3, and 6 allergens. In using the agrobacterium, we use its natural ability to transfer this plasmid or vector into our peanut plant's genome. One of its many advantages include low transgene copy number and the ability to generate lines free of long stretches of DNA with defined ends. It is the most reliable way to generate gain of function or loss of function mutants to assess gene function. Our desired gene is introduced along with our vector into our tDNA region specifically. Our vector can then become mobilized into our agrobacterium host strain, and that itself will act as a delivery mechanism that can physically introduce our DNA. The agrobacterium will also have to be used in the selection pressure. This is just a necessary step to ensure that only our desired genes grow into the peanut plant cells. After co-cultivation, explants are transferred containing our antibiotic hygromycin phosphortransferase gene. The hygromycin will kill all of the untransformed cells, and only the cells that were incorporated by our vector and the HPT gene can survive and generate. The viability of our plant will be very important after our delivery of our gene. We must ensure we introduce a good rooting system for vital survival of our peanut plant. This includes the moisture, temperature, light intensity, and nutrition of our plant to be monitored closely, just to get and make sure that our plant has good and viable seeds. We can also use a different method, which is our biolistic delivery. This would use a standard gene gun. This is another method that can be used to deliver our genes into our peanut plant. The gene gun has been used in the past before, but has had significant issues such as inefficiency, inconsistency, and tissue damage. However, this is different because they introduced a new flow guiding barrel, which is a new component that allows for nearly 100% of target control and particle control into the gene plant to better introduce the DNA and vector or plasmid into our peanut plant. This works by coating gold or tungsten microparticles with our vector into the genetic material and into our peanut plant cells. The cells that survive the particle introduction take in the introduced DNA and express the desired traits that are needed in order to target our desired allergen genes. These transformed cells can be grown genetically modified plants and into full-grown peanut plants. Our flow guiding barrel ensures that a much higher percentage of the microparticles can consistently enter the cells of our peanut plant, which can help them withstand the growth process, ensuring us a better genetic process.
So once again, to reiterate, what are we doing
So we are genetically modifying and removing the allergen from our peanut. So by this, we begin by the DNA structure manual from the plasmid or the vector. We target the selection through our major allergens, which are two main allergens are ARH2 and ARH6, as well as ARH1 and ARH3, which will then silence, which will altogether be silenced together by creating an immunoadjacency, which is similar almost to like a vaccine or a pathogen, which helps trigger an immune response with the body. We will separately be changing the genes, which is AHFAD2A/B, which is the oil contaminants, and we will be slowly reducing the oil quantity. And by starting off, we will be assembling the crisp Cas9, which includes the plasmid being engineered into two parts, the Cas9 enzyme gene, which will act as a pair of scissors, which will then guide the RNA, as we can see. And by guiding the RNA, they once again start as scissors, and we will then help, which helps pinpoint where in the peanut's DNA. This ensures the compatibility of the Cas9 gene, which is a bacterial. This is then placed under a plant promoter by the peanut cell machinery to actually read and act as like the instruction manual. This then engineers the plasmid to follow and clone itself to mass produce, which is then is created a specialized bacteria to purify in a larger quality to clone the DNA, so it's ready to be used. The second stage of this delivery is to make changes in which we specialize the techniques of the gene gun, which is the biolistic delivery. So by doing this, it's essentially a natural carrier from the bacteria, which is an agrobacterial method we also use on top of it. This helps knock out the peanut cell because of the Cas9 scissors are guided by the gRNA to target the gene, which is the sequence of it, and knocking out once again the allergy gene, which is a double-stranded break, a clean cut on top of it. The cell will then try to make the mistakes and deletions, but because of this, the indels will create like mistakes, and which helps them target the genes, and once again creates a better and modified version. This creates a more permanent knockout allele. And lastly, the final stages of the succession of the modification is for the survival and the growth of the plant itself. Now that the plasmid now carries the selection marker and creates a resistance to an antibiotic, now the delivery of the peanut cells are grown into a rough environment which contains the antibiotics on top of it. A few of the cells are fully added to the new crisp tools so then it will grow. The regeneration of the survivability and the modification of cells will then grow and through the tissue culture until they become newly genetically modified peanuts, which is its transformation overall. This will then cause the end goal of the peanut, which creates a pyramid deficiency in all the allergens. In a singular plant, this creates a lower allergen risk as well as maintaining its flavor and quality.
Utilizing the CRISPR-Cas9 with our vector helps us to disarm the peanut protein at the source of its allergenic start, so that the allergen is never produced in the first place. Using guide RNA and Cas9 enzyme, the guide RNA targets the allergen gene, and the Cas9 enzyme cleaves the DNA at that precise location. When trying to repair the cleaved DNA through non-homologous end joining, the cell will cut out nucleotides, disrupting the gene sequence and resulting in a gene knockout. This causes the entire allergenic protein to not be produced in the first place. This method provides a clean solution to this persistent public health issue, and the CRISPR editing maintains the plant's growth, yield, and flavor while only affecting the allergenic protein. It allows for a focused, non-invasive modification rather than extensive genetic overhaul.
As said previously, the CRISPR-Cas9 provides a direct, precise, and very stable solution by eliminating the entire allergen instead of targeting the proteins and still allowing the plant to further produce. It disables it at its source. In comparison to other methods, starting with RNAi, RNAi only reduces the mRNA allergen rather than complete removal because it does not lead to a full knockout. Other allergens are still produced, and other factors that play into the downsides of RNAi are tissue type and environmental conditions, which overall lead to an unreliable and sometimes varying effectiveness process. Another comparison to make is a post-harvest allergen removal, which focuses on heat, and the main protein allergens, which are ARAH2 and ARAH6, are very heat resistant. So a lot of these typical processing methods don't work, and once again lead to just a halt of processing, not a complete knockout. Another comparison to make is the random mutagenesis. In comparison with CRISPR-Cas9, the CRISPR-Cas9 does not create a loss of function or off-target mutation, and the mutagenesis does, which overall leads to unwanted factors in the plant or overall disruption of growth. And CRISPR is overall more effective just because of its specific cuts and its locations. It forces the plant to use deletions or insertions to then disable the production of the allergen in totality instead of leaving it just treated and allowing further productions. So overall it is a clean, predictable, and stable option. The few challenges come from technicalities, which are usually determinant from regional-based labs. So nothing with the CRISPR-Cas9 allergen removal method in itself, just environmentally based. And in terms of regulation and acceptance, edited peanuts by the CRISPR allergen removal are overall safer and do not contain any foreign DNA such as regular GMO methods would. So it leads to a safer option for peanuts because there's no outside DNA. Although there is different regulatory and filter barriers, that is really one of the only drawbacks.
While genetically modifying a peanut can be beneficial in many ways, we also have to think about the legal and ethical complications involved, starting with all the resources, tools, and the scientists that we will use to do so. We must think about the compensation that the scientists are going to receive and all the money that the tools cost. This ultimately leads to an increased price of the peanut, which is not well for the low-income community, as if the peanut is increased to a really high price, then the lower-income community will not be able to afford it, leaving only higher-making income people, the consumers. Another issue that we have to think about is that this peanut will go through many testing and it'll eventually be FDA approved. But what comes with that is the labeling. When you're selling a product that was genetically modified, you have to label it as genetically modified, and failure to do so can lead to legal complications. So it's really important that we think about the labeling. Lastly, we have to think about when CRISPR-Cas9 is modifying these genes, CRISPR-Cas9 isn't perfect and it can make some mistakes. So let's say if the CRISPR-Cas9 were to accidentally remove a gene that shouldn't have been removed, that can lead to other problems such as an overgrowth of a protein or abnormal peanut growth in general, or even a protein growth that has never even been seen in a peanut, and that can lead to long-term effects, and who knows what that will cause. So to prevent this and to deal with this problem, scientists must do long-term testing to see those long-term side effects, and they must test between children versus adults.
Now, as we have discussed, the goal of this genetic engineering project is to knock out the allergen genes, especially on genes like ERA H2 and 6, since we know these are the genes that produce the allergenic protein. This is why knowing the DNA and amino acid sequence location of the gene becomes important, since this constitutes the information map that the guide RNA is going to need to direct the CRISPR-Cas9 tool. And this is where the modification must occur, otherwise it would be impossible. Now, in conclusion, and all things considered, we believe that despite the skepticism and the implications discussed, this strategy would help remove one of the most common allergies worldwide, and it would make this genetic modified product available to the public.